Thank you, and thanks to to ARM for putting on this conference and giving us an opportunity to talk with everyone. So I'm the Chief Strategy Officer for Form. We're a bioinformatics and machine learning AI company working specifically on some problems in cell and gene therapy. What I'm going to share with you, what we've been working on over the last couple of years, and how we think we can help move this field forward. So I think we all know and we can all agree that we're hearing a lot about AI and machine learning and the potential for this to be really disruptive in our space. But what I think we also know and that we want to be careful is how do we use this effectively? How do we use it to address the right problems and to not kind of get caught up in some of these hype cycles that we've seen when other computational solutions have come to biotechnology? I always like to joke that like biology wakes up in the morning and says, how am I going to make it really hard for software today? And the things that are really important in biology have a lot of redundancy built into those systems. So we need to use these tools very carefully and make sure we're using it for the right solutions. One way to do that is to partner with somebody like Form. We feel like we're kind of a new contract organization that a cell and gene therapy company might work with, kind of a contract computational organization. Just as you work with somebody to help you outsource some of your research or outsource some of your manufacturing, we're a partner that can help you outsource some of your bioinformatics, your data science, and importantly, your use of AI and ML. Now, why would you want to do that? I think, again, the same reason you would do it for some of these other use cases that we know about. It doesn't make sense to build out an entire machine learning AI group when you're a small, nimble, hungry biotech, and every value inflection point is incredibly important. And building out this kind of functionality can be very difficult. And then other reasons that you might want to do this is you might want to augment. Maybe you do have some bioinformatics and data science, but you really want to bring in some expertise of folks that really work with a number of different clients across the space so you can get the breadth of their knowledge, the breadth of their experience. We also have been fortunate to partner with some leaders in this space. We're part of Google's multi-omics platform. We're actually their exclusive multi-omics platform, and we're able on our platform to use a lot of the really cool tools that Google is now developing in this space, like Deep Consensus, Deep Variant. And so that's, again, a very important capability and expertise that you can get access to. PacBio, we exclusively are their partner for analyzing the data that comes off their long-read machines when we're analyzing your drug product. We'll talk a little more about that. And NVIDIA is one of those companies, like 2 years ago, I'd never heard of it, and now I can't not hear about it. You hear about them all the time. But we're working with NVIDIA using their Parabricks platform to be able to do these kind of computational things way faster. And because we're doing it so much more quickly, we're able to do it much more cheaply for our customers as well. So by partnering with these really leading technology companies and sequencing companies, we're able to help you get your products through the process faster and more cheaply and then more safely, I think is an incredibly important point as well. We're also working with a number of academic partners and making sure that we're bringing in those leaders in this field to inform how we're trying to solve these problems. Now, an interesting thing, we are hearing a lot about AI in drug discovery, and I think that's an incredibly interesting and important space to look at. But one part of that's a long time to capture that value that you're creating in the AI drug discovery. We're focusing a little farther down on that value creation chain. We're really looking at that preclinical development. Kind of once you've found something that you think has potential to become a drug, I kind of have an analogy to the hit to lead in the small molecule development path. When you have something, you have your gene of interest, you want to go after, you have kind of some preliminary proof of concept data in an animal model, we can work with you to help develop what that is going to be as far as a drug. So how do we optimize it? How do we make sure we're asking the right questions to say that this is something that is manufacturable, that this is safe, and that this does not have truncation hotspots, for example. So we're really focusing on that part. What I like about that is it's been a little bit scientifically de-risked. Nothing is ever, completely scientifically de-risked. We all know that. But it has been scientifically de-risked a little bit. But there's still a lot of operational risk that we can help you address. And that is a faster path to capturing some of that value. Now, the way we do this, and we'll get into some more of the specifics, but again, I think something that's incredibly important when you're thinking about how to incorporate these kind of technologies into your development plan is how are we training our models? And so we're training our models. We have a number of data sets that we've gotten, either public data sets, but we also have a number of proprietary data sets, either from early customers that have partnered with us, or we're actually sequencing enormous amounts of gene therapy drug products from our academic partners in order to train our model. And then we are doing biological validation and taking the results of that biological validation and putting it back into our model. And that's an important part of the feedback loop. I think whenever you're using these kind of tools, if you don't have that validation step, you always run the risk of bias. And I think there was an article just this morning talking about, it was a clinical example of using AI, that the more you use it, the more the bias kind of gets incorporated, and then you end up with data drift. So we're being very intentional about keeping that data feedback loop very, very robust so that we can reduce the risk of these kind of bias events that happen in this space. So when we work on this, we can be your in silico partner. So when you have your scientific proof of concept, you have your therapy that you know works, you can come to us and we can model a number of different things using our incredibly robust models and help you ask some questions about what should that next step be. And so we can take, if, for example, you do have a drug product and you have some long-read data, we can help you analyze that and tell you what's in there. Are there chimeric molecules? Are there, host genome? Do you have truncation hotspots? And again, I think that goes back to how quickly this field is evolving. It was just a couple of years ago that the FDA was starting to ask about, we want to know about the empty to full ratio, and now they're saying we need analysis of what's in your full capsids. And so, we can help you understand that from your long-read data, so you can understand what are my contaminants. That's going to help you with your dose calculations, also help you with safety. So that's important. We can simulate your bioreactor runs. We can help you understand what's the best molar ratio for your helper plasmids. We can help you understand when should you end, when is the peak of your capsids being filled with drug product and not filled with some of these contaminants. We can also we call it multi-candidate comparison, where we can model, taking your gene of interest, we can model 8 different promoters that could be potentially tissue-specific in the appropriate strength, plus and minus introns, different poly-A tails, and let you know of all of those, which is the one that has the best manufacturability. Which one are you going to get the most capsids out of your manufacturing process that are filled with therapeutic product? And, that's an incredibly important thing. And I think, again, this is to augment what scientists can do. I think, another thing that we have to remember in this space is that it's important that we don't ever think that we can do this without the creativity and cleverness of a scientist. So these are tools, these are things to give the scientist a smaller world of opportunity to look at to inform that next step. So we have a number of ways that we can help you with this step as you translate into the translational path, the drug development path of your drug development entire pipeline, and always trying to keep your outcomes in mind and at the forefront. So whatever your problems are, let us help you try to come up with ways to model it and understand what should that next step be using the power of the computational tools that we've got today. So this is just an example of that multi-candidate comparison. This is one where, for a customer, we looked at, I think it was 9 different promoters. Every promoter, we did plus and minus the intron. We had some different regulatory elements in there, different poly-A tails, to say which one has your best likelihood of getting full capsids, but then also making sure that those capsids when you use them in a clinical setting, that you would get mRNA production, and that mRNA production would also make protein expression. Because again, every one of these steps has the opportunity to go sideways, and so we can model every step along the way to help you figure out what the next thing is. So we've been in existence publicly for about a year. We were incubated in stealth for a few years before that within another molecular biology, synthetic biology company. And so we've been talking with a lot of cell and gene therapy companies over the last few years. I'm not going to go into all of these. We have a booth just outside the main conference center. We'd love for you guys to stop by and talk to us about your specific problems. But I am going to tell you some of the things we've learned from our customers and how we've helped them solve these problems. Importantly, biological validation is key. Again, I think as scientists, we come to the table very skeptical, and so we want to make sure we address that skepticism head on. And so we have been doing a lot of work generating our own biological validation data, as well as back testing with known data, and working with NGS data that we get from our customers. And just a quick example of that, this is one where we were able to model a customer's construct. This is one that is far enough along in the development process, there's not an opportunity to change it. But what we were able to do was take their sequence, model it for where there were truncation hotspots, and then actually take their NGS data and see how it matched. And what we can see here is we absolutely were able to, the NGS data is the green, the blue data is the original construct, and as you can see, we were very, very good at being able to identify where those truncations were going to happen. And so, again, important to understand what's in your drug product. Now, this is one that's far enough along that there's not an opportunity to change the sequence, but we did run our AI, which goes beyond your traditional synonymous codon substitution, but looks at codon substitution, holding GC content consistent across it. And you can see in that orange line, we do predict that if we were able to change it, keeping the protein product constant, keeping the mRNA and protein expression constant, that we could potentially reduce the truncations in that and getting a purer, more potent product. Another thing, lack of standardization. We heard that this morning from the gentleman from Pfizer. Anytime you've got a new modality like this, things are moving very, very quickly. And so, there's a lack of standardization. So we're trying to be part of that solution, and we are working with the academic leaders and industry leaders in this space to really drive kind of consistency in the characterization of those long read sequences. And what we can show from that is we can give you a report that tells you what's in your drug product, how much of that is the full genome, how much of it are partial genomes. In those partial genomes, what are they made of? And we've already had one customer use that as part of their IND meeting for the CMC portion and get no pushback from the agency because they were able to tell, we do not believe that there will be any weird proteins made from this because we don't have promoters with open reading frames that are not therapeutic, for example. So that was incredibly useful, and that's really important for those conversations with the regulatory agencies. Finally, what we started with was just this idea of how do we take the gene of interest optimization and take it to the next level using AI to make it a better drug. And what we found is that's not enough. They want a lot more. And so what we've been able to do, we've been working with another publicly traded gene therapy company, and we've been helping them characterize their drug product, but also run those multi-candidate comparison reports to help design the parts of their pipeline. And then we're using all of that information, layering on generative AI to help them develop their next generation of products. So these are all ways that we would love to help you move your pipeline forward and understand what pain points you've got so we can bring the right tools and expertise. We have one of our AI experts here, which I will be the first to say is a little magical to me. So if you have specific AI questions, we have someone who can answer those for you. But we'd love for you to stop by and talk with us. Thank you.